Thank you for the invitation. I'm really happy to be presenting the work. I really wish I could be in Europe and visiting and doing this in person. But I guess in this day and age, this is already a lot of fun to be able to join from all the way and without having to travel. So today, what I'm going to try to do is to talk a little bit about some of the ideas that I discussed in this recent book that I wrote that is called The Entangled Brain. The book itself was written with a very broad audience in mind, not necessarily specialists like in the group here that I'm talking to. So I'm going to be describing some experiments, the actual work that I've been doing that connect and have inspired some of the ideas I wrote in the book, but I'm going to be staying at the level of the experiments and the actual research, and so not as informal or as maybe speculative as the book is, because in that venue we can do different things. Okay, so with that, let me get started. So I've been interested in emotion, the brain bases of emotion, for quite some time, and also really interested in understanding how the ideas have evolved throughout the past century or so. And if we look at the literature, we really see that historically, the idea has been that emotion involves a separate and independent circuit. And the circuit in the brain has changed through time, but perhaps one of the strongest legacies and one of the most influential legacies has been of this idea of a limbic brain, a limbic system, a visceral brain, that was proposed in the late 40s by Paul MacLean. It was a circuit that was really interesting because it was in the 1950s, but it was already the idea that it was not about a region, it was actually a circuit. The circuit was centered on the hypothalamus and the hippocampus and the cingulate gyrus, the cingulate cortex, and so on. But the idea is that there was this specialized circuit for emotion. If we fast forward all the way to today, the circuit now obviously is going to be looking very different. This is like 70 years later. We have learned quite a lot more. But the notion of a dedicated system really still plays a central role in our thinking. How do we think about the brain and a faculty, a domain, a mental domain such as emotion? The emotional brain comes in many different shapes. If you ask maybe a dozen people that do work more closely to this area, they will give you slightly different answers. But as I think it's fair to say that there are many regions that are repeatedly discussed and included in this so-called emotional brain. Regions such as parts of the cingulate cortex, especially the anterior cingulate cortex, the orbitofrontal cortex, the anterior insula, parts of the anterior lobe sometimes are included. And there are other parts of the brain that are definitely included, including the amygdala and the hypothalamus as it used to be also included, but less so now, and more the emphasis on the amygdala, perhaps a more amygdala-centric view. So in this notion of having a dedicated circuit for emotion, there's another theme that is really important to consider, which is there has been the tendency to have, or the frameworks have been centered on a specific structure, in a brain structure, in the sense that there is the dedicated circuit that is not only a dedicated circuit, but there is a region within the circuit that really plays a disproportionately important role. And as I mentioned before, throughout the 1920s to 1950s, going all the way to the 1950s, this structure was the hypothalamus, a region in the basal forebrain that is incredibly multifaceted, does a lot of things, but was thought to be incredibly important for emotional processing. And if we fast forward to the 1960s, and then later establishing itself in the 1990s up to now, then the amygdala, another basal forebrain subcortical structure, is really thought in many ways to be sort of like the center, the epicenter of this emotional brain. So this is a traditional view that doesn't neglect that there are other areas, but that really focuses on these areas, these or other areas specifically. So the claim that I like to make is that if we look at anatomical connectivity in the brain more generally, it suggests that we might be looking at this type of system from the wrong angle. If we consider for instance, the amygdala, more specifically the basolateral amygdala, the basolateral amygdala, it really is massively interconnected with much of the cortex, with really strong connections to frontal, temporal, and cingulate cortices, and a little bit less so to parietal cortex, and some connections to occipital cortex even. But the point being that it's really a massively connected region, and some anatomists have estimated, these estimates are based on rodent work. It's slightly different architecture, but in any case, it's a structure that might have as many as 600 to 1,000 connections. So the point that I'm going to be making is that we should be looking at these large scale interactions and looking at these large scale systems, as opposed to isolating, carving out pieces of the brain in terms of any given system. For instance, the emotional brain, which is something that I study quite a bit. This property of quite considerable and massive connectivity, as I was illustrating from the amygdala, is not just something about the amygdala. In fact, computational analysis of anatomical data, this paper that I quite like from Moda and Singh from 2010, that clearly needs to be updated by now, but even then collating hundreds of anatomical studies showing that there's really an incredibly exuberant and rich backbone of connectivity linking any given lobe or segment of the brain to other lobes. So for instance, from temporal cortex, you can get to the insula, from cingulate to parietal, from frontal to occipital, and so on and so forth. And obviously the incredibly important role of the thalamus and other subcortical structures, including basal ganglia loops. I should say that I'm more than happy for anyone to interrupt at any point and ask questions. I don't know if you have a preferred format that you want to reserve it to the end, but to me, it's either way. If I don't see you here on the screen, please, maybe Sammy can just say something, and I can just take the question. I'm happy to do it in whatever format you prefer.
Yes, please. I could read the chat and give you the message.
Okay, so I might not be seeing the chat though, so from my screen. So I don't see, like in front of me, I only see my own screen. So if you could just interrupt me and say there's a question, that would be helpful, because I don't see the pop-up of the chat.
Yes, of course.
So, okay, so having this in mind, one of the things that we've been doing in my lab is centered around an integration framework that, again, was inspired by findings such as this anatomical ones and by a lot of functional data that tries to look at aspects of perception and cognition that we typically tend to separate as deeply interactive, but furthermore, interacting with these separate domains that sometimes we put on the side, the affective emotional domain and the motivational domain. And this is actually something just here outlined for simplicity as two separate independent systems. I don't mean this, they're heavily interconnected, so I don't mean to separate them too strongly, but essentially by affective, I mean something as more negative emotions, withdrawing from aversive types of emotional content, and motivational significance here, just for convenience, I'm more appetitive, reward related. So we've been within this integration framework, we've been looking at a series of experiments trying to understand how these different domains, they might interact and in fact be more strongly intertwined. So let me illustrate a couple of those today. And I'll start with the question that I started, in fact, in the early 2000s when I was in Leslie Ungerleider's lab at NIH, where we were really interested in understanding the interactions between visual processing and affective content. And this was a study that I did a few years after leaving the lab, but very much in the tradition, in the spirit, in the types of thinking that and experiments that we were performing at my time there. So the experiment that we did was a variant of the so-called attentional blink. Some of you might be familiar with it, but let me remind everyone about what the attentional blink is. The attentional blink is a task in which over a period of around 2 seconds or so, the participant looks at a clip or a series of frames presented each for 100 milliseconds or so. So this is, you see my cursor when I move it around, right?
Yes. Yes.
Yeah, oh, great. So each frame here is being shown for 100 milliseconds. Then it goes to another frame, and so on. The point of this attentional blink task that has been studied probably by now, I would say even more than, it's not even hundreds, it's probably thousands of studies, believe it or not. I think that, yeah, it's very large literature. The critical element, there are two critical elements in this stream. One is called target 1 and target 2, and they can be very different things depending on the study. In our study, the first target was a face. The person had to indicate which face this was, for which they had been familiarized in the beginning. So they knew that there was someone called Andy, someone called Bill, and someone called Chad. It's a very easy detection task to detect this first and correctly name this first stimulus. In our task, I think they were correct 97% of the time. But the attentional blink refers to the fact that 200 to 300 milliseconds after the first stimulus, the first T1, appears a T2. And in this window of time, this T2 is quite difficult to detect. In fact, it's as if the person is physically blinking and not seeing and missing the stimulus, but it's not a physical blink, it's an attentional one, so-called attentional one, in which the person seems to be unaware of the second target in these streams if it's followed by the first target within a certain narrow window. And the idea is that there are many, many ideas, it's still being worked out, is that maybe processing resources are consumed and putting V1 in some short-term memory so that you can later report if this was Andy, Bill, and Chad. And then those resources are not available when you literally miss T2. T2 in this experiment here is an outdoor scene. It can be different things. In an outdoor scene, because we did an fMRI experiment, I'll show you why in a second. It's an outdoor scene. It could be, for instance, a house, a suburban house here in the US, or a skyscraper in a city. And the person had to indicate at the end of the 2 seconds what T1 was and what T2 was. And so what we thought was, what if, so the T2 is literally missed a considerable number of times. That's the attentional blink phenomenon. So that's the perception. I'm indicating here that their perception that even when a house is presented, they don't see it. They report not seeing it. They miss it. So what we thought was that, what if we add affective significance to the T2 stimulus? For instance, by pairing houses with mild aversive shocks during a session prior to this attentional blink session. If we do that, the stimuli will acquire affective significance. And does that overcome the attentional blink? Is it the case that it has stronger potency so that it could overcome these whatever bottleneck or competition that is happening here in the phenomenon, in the attentional blink phenomenon? So what we did, as I mentioned, we created 2 categories of stimuli so that a person could either see houses or buildings, one of which was paired in prior to the task itself in the scanner, but prior to the actual main runs. They viewed outdoor houses, they view outdoor scenes containing houses that were paired with shock. That was a mild shock calibrated so that it was tolerated, was highly unpleasant, but tolerated by the individual. And the other category was, there was received no shock. So we had a CS plus and a CS minus and this was counterbalanced across participants. So what we found was that affective content behaviorally reduced the attentional blink. In the actual normal category, meaning normal, not effectively significance, the attentional blink was fairly substantial. It brought performance to around 60 something percent. So this was a really challenging, task, but it improved by around 10% if the participant was being exposed to the CS plus category. And we did a series of analysis to look at whether there was changes in response bias and other kinds of things that we can discuss later to really have a better sense that was increased sensitivity and not something that people were more conservative or liberal to adopt one of the two categories. The experiments that I'm showing, the results that I'm showing here, and these behavioral results and the fMRI results that I'm going to show have no physical shock itself. The physical shock was used only in the beginning of the experiment to create this emotional affective significance for one of the categories. But the actual experiment is done under what's called extinction, meaning that the shock is not presented. We have intermittent short little sections where we boost up again the affective significance that the stimulus has, but it's not part of the attentional blink. So there's some kind of trick there to maintain the affective significance of the stimuli, but the task, the attentional blinks task itself, there's never a shock being administered. Okay, so what we're trying to do, we're trying to understand a little bit what are the potential interactions that these threat-related stimuli might exert their effects such that they have better performance. So what we did was that we knew that the amygdala is really critical for this acquisition of affective significance, that the stimuli became more powerful for the CS plus stimulus than the CS minus, as indicated by the behavioral data. So what we did was that we looked at the responses on a trial-by-trial basis, and we classified stimuli at trials in terms of whether they, for instance, were a hit trial or a missed trial. A hit trial, they correctly indicated that there was an outdoor scene, a house, and a missed trial, the house was presented, but they actually missed it. And so when we look at the trial-by-trial fluctuations in the amygdala, what we saw is that it was actually directly correlated, in a sense, predictive, correlated to behavior, such that as the fMRI signal increased on a trial-by-trial basis, we could more clearly, given in a logistic regression, we could more easily differentiate a hit versus a miss. At the hit versus miss proportions and rates were well fit by a logistic regression that as a function of the fMRI signal. In other words, the stronger the signal in a given trial, the higher the likelihood that the person would detect let's say the house stimulus. We created the house and building stimuli having in mind looking at a potential interaction between the amygdala and the parahippocampal gyrus, a region of the brain in anterior visual cortex that is very sensitive to these outdoor scenes and responds very strongly to buildings and houses and so on, and responds rather weakly to face stimuli as an indication, as an indirect indication, proxy for the extent to which the T2 was being processed. So the model that we wanted to evaluate then was, is this relationship between the amygdala and behavior actually mediated by the responses, again on a trial-by-trial basis, in parahippocampal gyrus that we could actually isolate via a separate localizer run. So what we found is that when we performed this analysis formally via a formal mediation analysis, the link between amygdala and behavior is actually explained by the link between the amygdala, the link between amygdala and parahippocampal gyrus, and the link between parahippocampal gyrus and behavior. So suggesting that a potential mechanism by which increased affective significance enhances perception is via these interactions. Obviously, this mediation analysis is not causal in any sense. It's just a set of correlations that are consistent with these relationships, but it's something that the prediction would be then that we could manipulate responses in these regions. For instance, if we could stimulate the visual cortex, we would actually find, we actually would be able to affect behavior in the way predicted by this little schematic model here.
Sorry, Luis.
Yeah.
Someone in the public is asking us, Damian, is this DCM is effective?
Yeah, no, we didn't use a DCM model per se. What we did was just a standard path analysis by looking at the trial by trial fluctuation. And basically the path analysis, then you can estimate these path weights based on that. So it doesn't really have, like in the DCM case, something explicit directionality of the connections or something like that. We tested a few alternative path models to see if that it was still consistent with this general idea. But as I mentioned, this is a suggestion I view as consistent with this kind of mechanism, but it's obviously with fMRI, it's not being done at a mechanistic level. It's really to provide hypotheses for the manipulations and to try to unpack some of the possible ways by which this increased affective significance enhances behavior.
And in the same way, there is also some Wagner that say, is there more evidence from various affective stimuli other than the presented houses that visually are more similar? To say that.
Oh yeah, I mean so for instance, there's again, this literature is so incredibly enormous that is so almost embarrassing. But for instance, T2 is often a face. T2 so for instance, they have different T1s and T2s. Sometimes T2 is a face. For instance, a T2 could be an emotional face. Could be, well, let me put it this way. A T2 could be a face with an emotional expression, a happy face, a fearful face. And people have claimed that a fearful face overcomes the blink a little bit better than a neutral face. People actually, you can take a fearful face, a neutral face, and condition it with pairing with shock, and it does the same. So there's a very large literature looking at both in the appetitive domain, reward related, and the aversive domain. There's a very broad range of stimuli and manipulations that have looked at how this competition between T1 and T2 is balanced by, I should say, the potency of each one of these stimuli. So one way in which it could be more potent is that it's very bright and moving stimulus, or it's effectively significant, or is linked to reward. Can attentional modulation without affective modulation have an equivalent performance? Yeah, so I think people have done all sorts of attentional types of manipulation too. For instance, I'm trying to see spatially priming that. The attentional manipulations I can't remember right now, but for instance, with reward, it also works. And with other features that are important for spatial attention that highlight that T2, for instance, if its border suddenly becomes a little more stronger, more brighter, I think it helps it compete. So it's a really general phenomenon that these T1 and T2 compete, and their effects of T. In fact, if you change T1 in certain ways, it will also increase or decrease the blink. So it goes both ways. There's a really fascinating literature there. Okay, so I still see something. Ah, yeah, thank you. So many of these ideas of the enhanced processing of affective stimuli have suggested to some investigators, at least, that the processing might be even in fact automatic in a way that it would be advantageously evolutionarily speaking. For instance, a person might be trekking in the desert. This is a slide. This is a figure from Joe Ledoux. A person is trekking in the desert, is not paying attention to that rattlesnake, but the rattlesnake is in the field of view. It's being processed, and it's processed fast in an automatic fashion. So the person turns around and runs, and it's saved. So this kind of affective significance evolutionary link has been highlighted by many people, to the extent that it's been suggested that this idea that there's an evolved module for fear and fear learning is really something very central to the organization of the emotional system in the brain and behavior, giving the animal preparedness, but at the same time, possibly being if it is overworking, might be leading to phobias and fears. So these ideas led Ralph Adolphs and I to write a review paper a while back, in which we coined something that we call the standard hypothesis. The standard hypothesis is the idea that emotional stimuli are processed by a dedicated sort of modular system that operates really rapidly and automatically. So there's no need to pay attention, and it might even be without awareness. The defects in this system might be even more than just something that we should study from a basic perspective, because defects in the system have really important clinical implications, including perhaps influencing phobias, mood disorders, and PTSD, post-traumatic stress syndrome. So in my own lab, and also in Ralph Adolph's lab, we were looking at these ways in which emotion laden stimuli, affectively significant stimuli, would be influencing cognitive operations and cognitive processing in general. So to test the idea about the extent to which they might be this evolved module that operates independent of attention and awareness in a completely separate, powerful way. So in both labs, we did lots of experiments involving awareness, involving attention, involving contextual manipulations, to try to understand the extent to which we can really say that this happens in a largely automatic fashion, at least in the standard automatic type of lacking independent of attention fashion. So let me just very briefly provide an example of the study that I did long ago. This one was actually when I was at NIH. We were interested in this and many other studies that we did after this in the role of attention. So how much paying attention to a stimulus plays a role in the processing of this category of stimuli? So in one block of trials, a participant looked at the screen with these stimuli here and had to attend to the faces and just indicate whether they were male or female looking. And it was an easy task. People had no trouble. These were cropped hair and strange Ekman faces, so they were not at 100%, but they were 91% correct. In alternating blocks, people actually they maintained their fixation, their gaze fixation that can be controlled. We didn't do it in this experiment, but in subsequent experiments did with eye tracking and whatnot and better designs. But in this case, they maintained their fixation and just moved their focus of attention to the periphery to answer the question whether these peripheral bars were of the same orientation or different orientations. And this was designed to be very challenging, to really require quite a lot of focus and carefully moving their attentional focus. But the idea here is that the visual stimulus is exactly the same. We're just manipulating attention. And in this case, the stimuli were presented for 200 milliseconds. So by moving your eyes, by that time, the stimulus is kind of gone. So it's not really advantageous to really move your eyes. But as I said, one can easily do, and we did later, eye tracking. And this task was quite difficult. People really struggling with this task, with saying whether it was possible was quite difficult. But what I want to just briefly show here today is just one of the results, which is, so what happens to responses in the amygdala in the face of this attentional manipulation? What we found was that when the person is looking at the faces, when the person is attending to the faces, not surprisingly and consistent with what had been reported many times by that time, was that fearful faces evoked quite a bit of a strong response here, fear-attended faces, and much stronger than neutral faces, something that had been shown by in several studies before us. And we also saw an intermediate response for neutral faces, for happy faces. The question then, the contribution of the study was to show, okay, what happens when you really deplete attentional resources? And so what we found was that when you depleted attentional resources, there was no differential response as a function of stimulus type, fear, neutral, expression type, fear, neutral, or happy. So based on this study and a series of other studies from my lab and then several other labs, it really suggested that the effect of valence is strongly dependent on attention. There's a strong attention or cognition by emotion interaction in that the effect is only expressed when you're attending, but not when you're not attending. So this is just one example that I wanted to illustrate that led us to the conclusion in that paper, and something that, based on a much larger literature that we review, that yes, true, affective processing is prioritized. In fact, I just started the talk with the attentional blink, which is exactly an example of that kind of prioritization. And we were really interested in understanding the mechanisms by which it's prioritized. However, but it also depends on multiple factors, including attention, awareness, context, and so on. We're not showing a host of experiments that Ralph and others did on awareness, but even our study of the attentional blink is such that in the CS plus category, people still had a blink. They detected T2 72% of the time, instead of close to 100% of the time. It was much better than the CS minus, which was at 62. So it was a 10% increase in performance, but it was not something that people could detect the stimulus or evoked amygdala responses in the absence of perceiving it. Okay, leading to the conclusion that affective processing is indeed prioritized, but it's not strongly automatic in the sense that has been proposed in the literature that perhaps even supports clinical conditions such as PTSD and phobias. Okay, so let me shift gears now, and let me just make sure that I'm not going too slow. Okay, so
sorry, we have another.
Okay,
question
sure, no problem.
For Sophie Benitez, she said, can more extreme emotional stimulus still induce a response after attentional blinks?
Yeah, so that's one of the problems that we have when we study emotion research in the lab. We obviously have to operate within ethical grounds and ethical parameters, obviously. So there's only so much that we can increase the strength of the stimulus. So it would have to be a more, maybe VR, more immersive, could be more potent. Yeah, I think that, so I think that there might be manipulations that are more powerful than just pairing with shock. Is something that you experience much more embeddedness of yourself in a VR world. Yeah, that's true. I actually don't know. I should look into this. I don't know if people have used, I'm sure they have, VR for these kinds of studies. Yeah, it's interesting.
Thank you. And also a question.
Yeah,
There is
sure.
some like a ethnicity difference in this type of responses?
We never looked at cultural differences, and we never looked at as a function of for instance, manipulating the types of faces or anything like that. Like there are a lot of studies with, let's say, white participants and black faces, black participants and white individuals, and we haven't done those. Yeah.
Thank you.
So let me shift a little bit to the other side of this equation, because I want to also illustrate these interactions in the appetitive domain when we manipulated reward. So the hypothesis that we had was that motivation, as manipulated by reward, would enhance executive function, not in the sense of making you really fast or kind of overall energized, but actually would provide a selective advantage to cognitive processing. In this case, the selective advantage, the specific advantage, was in selecting the part of the stimulus that was more spatially selecting the part of the stimulus that was more relevant. So to do that, we investigated a version of this Stroop task in which there is an incongruency between the stimulus mapping, the mapping of what you have to respond, and another feature of the stimulus. So you have to respond what the color of the ink the stimulus is, and the color of the ink is red, but you see the stimulus and you also read it. After 20, 30 years of exposure to reading, that's fairly strong and automatic, very, very potent type of operation. So that generates a response conflict. So the way we did that, because we had in mind going to the scanner again with these kinds of stimuli, houses and word stimuli that evoked relatively responses in relatively separate parts of the brain that we can use as an index of the extent to which they are responding. What we did was we had these scenes and people had to indicate whether the picture contained a building or a house and just ignore these words or these strings on top of them. So those are task irrelevant. They were told just ignore that. And so then we had the congruent stimuli and the incongruent stimuli too. So it's essentially a very basic variant of response conflict. And so what we did, we ran this task with a manipulation in which was a reward condition versus a control no reward condition. The trials were exactly the same except for this initial cue that indicated what condition the person was in. And the target stimulus itself came a few seconds later. And again, were those house buildings that the person had to just ignore the words. So let me just briefly show you what we did, what we found behaviorally. So behaviorally, what we found was a reward by cognition interaction, in that the interference effect, which is the slowing down when you have an incongruent situation, when you have, for instance, a building, but you write house, that slows you down. This interference effect was reduced with reward. And so what I'm showing here is a comparison between incongruent trials and neutral trials. Neutral trials have just these X's. Instead of having something congruent or incongruent, we have a neutral condition also, which is very useful to try to separate interference from facilitation, right? Because there are these two possible ways in which these two dimensions of the stimuli operate. And in many of these experiments, it's very useful to use a neutral condition. So what we see is a clear slowing down when it's incongruent, when there's no reward involved. When there's reward involved, there are two effects. An unspecific one, which is people go a little bit faster. So notice that this scale here is truncated in the y-axis and starts around 500 and something milliseconds, is not starting at 0. So it's not that people are going super, super fast. They just increased around 50 milliseconds or so of their in their response time. So it's not like they're at floor and just going full blast. They went a little faster, but what we see now is that there is a decrease in this interference effect that we observed before in the rewarded condition. And we can also do some analysis to eliminate, again, that there's some kind of floor effect, that people are just going really fast, and you can try to really look to really understand that this is a more legitimate interaction pattern. So in terms of the overall model, what we were interested in testing in the paper was this idea that how are these interactions taking place? So we have a cue stimulus being shown here. And at the time of the cue, we were interested in understanding how interactions between regions that are important for top-down control, for instance, in the parietal and frontal cortex, are perhaps functionally interacting with regions that are important for reward-related processing, for instance, in the ventral striatum. And we were interested in understanding how these changes that might be happening at the time that the cue is presented impact in the performance a few seconds later. Remember that this happens 2 to 6 seconds later during the target stimulus, when I have to indicate that this is actually a building. So during the target phase, what we have is two things. We have a stimulus, and we can look at visual processing to look at the extent to which there is distractive processing that is related to this word. But we can also look at, during the target phase, at regions in the medial prefrontal cortex that are quite sensitive to response conflict. And so we can evaluate the extent to which this top-down cue processing affects distractive processing in visual cortex, and thereby potentially reducing response conflict in medial prefrontal cortex. So this is actually what we found. So if you find this interesting, you can take a look. But given time, I just want to briefly not focus on the entire study. I just want to briefly focus on the cue phase itself. So the idea here is to try to understand, is something in the brain happening so that the brain is reorganizing during the perception when perceiving this cue, so that it prepares you to perform this later target task a few seconds later. So to understand that, what we did was we took, again, it was during the cue phase that this was what we were focusing on. So we isolated the response to the cue. We can do that by doing some multiple regression and including the regressors for the target and the cue and separate them and whatnot. So we looked at the interactions. We were interested in looking at the interactions between regions that were important for attention, such as I mentioned, the frontal and parietal cortex here, the frontal eye field, and the intraparietal sulcus, and regions important for reward-related processing, such as the ventral striatum. And what we were trying to do is look at the fluctuations on a trial-by-trial basis of the responses in the frontal parietal cortex and the fluctuations in ventral striatum in those same trials. And what we found, or we wanted to evaluate, was to determine how this functional coordination or functional correlation or functional connectivity between the frontal parietal cortex and ventral striatum was affected as a function of reward. And to do that, we just run a functional connectivity analysis. Functional connectivity analysis is essentially the idea that you can take the time series of data or trial by trial responses and correlate, and you get an index of the extent to which these brain regions might be functionally coupled. This is not something that depends on strictly on having direct anatomical pathways. It can be via indirect pathways. The idea is that this provides an index. We can discuss what we can really get out of functional connectivity later in the discussion period, but that was the index that we use here. And what we found is that this functional connectivity between these regions actually increased with reward. During the reward condition, the functional connectivity was significantly stronger during the reward versus the no reward condition. But what we see here is that we see these functional interactions, this functional connectivity between pairs of regions. So what we wanted to do was to go beyond pairs of regions and go to the network level. So what we performed was a simple network analysis in which we took all the brain regions that had a main effect of responding to the cue, irrespective of whether it's a reward or a neutral cue. And we took these as our regions of interest in our network. And we built a simple graph with these brain regions. And we took the functional connectivity between these nodes during the cue period to be these trial by trial estimates. We estimated it on a trial by trial basis. We can do that. There are good methods to do that. And so we use those fluctuations on a trial by trial basis, correlating them, and you get the strength of the functional connection. When we do that, what we did was we started doing that. Again, just reminding you that this is only for the cue phase. So we're trying to see how it reconfigures during that cue phase. What we find is that the network can be well described by two clusters or communities. So a cluster of regions that are more tightly correlated with each other, that is mostly of cortical regions like the frontal eye field, the parietal sulcus, the anterior insula, and so on, and a region that includes subcortical regions, including the caudate, nucleus accumbens, putamen, and some other regions. There's nothing inherently correct or fundamental about this organization, in my view. My view is that you're just finding one way in which you can cluster these regions under the no reward condition. Because what we really wanted to do was to look at what happens in the reward condition as a contrast. Let me take this question before I show this result. Oh no, we did not explore something more higher order organization, or it was just very basic, just very basic organization that we looked into. Because I'll show you why in a second. I mean, or at least part of the reason why was because what we really wanted to do was not to characterize this organization here during the no reward as something, saying that there's something important about it. What we thought was important was to understand how the functional connections between these two clusters change when you have the reward condition. So when you do the reward condition, when we did the reward condition, what we found was that there, I don't know why this is not, my mouse today is misbehaving a little. Yeah, it's not showing exactly the way I wanted to show, but that's okay. What I wanted to show was that the connections in purple here are the ones between the two clusters. And what we see is that functional connectivity consistently increases between, they increase a little bit within each one of the clusters, but they considerably increase between the clusters in a way that if you use quantitative measures such as global efficiency or modularity or other things, you really see that this overall system becomes more efficient or less modular. So in other words, this system that is organized during the no reward condition that has some functional interactions linking these two clusters, it becomes much more integrated during the reward condition, showing these kind of large scale changes in network organization as a function of experimental condition reward in this case. So these changes in functional connectivity are happening from the standpoint of a given region. It's changing its functional connections to lots of nodes, not just a pair of nodes. So we wanted to contrast that to the situation in which we typically might focus on changes in functional connectivity between a couple of regions. So let me just show you one more study before I wrap up, but I'm just going to show you just rather quickly, because what we wanted to understand was how do these large scale network interactions that I just showed you in the case of reward might be also be happening in the domain of threat processing. So what we had was a study in which people were in two kinds of blocks. The blocks were were around 60 seconds on average, with a lot of unpredictably of how long the block was. And the safe blocks was just essentially resting state. I mean, don't do anything. There's nothing to do. It's a safe block. It's indicated by this blue circle here. You're never going to get a shock, and you never do get a shock, so it's safe. In an alternating fashion, you also experience so-called threat blocks, in which you might receive 0 to 4 shocks at random points of this block. So you wanted to create a block in which you kind of had some kind of anxious apprehension throughout this block, and you couldn't predict how many shocks, and even if there were going to be shocks. And so we want to contrast the organization, the functional organization under these two conditions. And I should, again, reminding you that the trials that have shock are there just to make you believe that I will use shock, but the trials that we want to evaluate, to study, don't contain shock, because we want to just study anxious apprehension and not the shock itself. So we want to use shock as an instrument, but not as the object of the study itself. So what we did was we took these large-scale networks that are well known in the brain. Let me see, there's a question here. Yeah, let me take that one a little bit later. So we looked at these large-scale, we wanted to look at these large-scale networks, salience network, executive network, and default network, and things like that, and very well characterized in the literature. And what we did was that we took some measures of network efficiency, global efficiency that we can discuss a little bit later if there's interest, but they're a very large class of them. This one is just an interesting formalism that considers both direct and indirect paths that to compute both efficiency, the extent to which information spreads or at least has the potential to efficiently spread across the network, and so on. And again, the nodes are the time series correlation that from the task in which the person is basically doing either resting state or sort of like an anxious resting state. What we did then was to take regions from 3 large networks, salience, executive, and default networks, and we added a couple of regions that are important in anxiety and fear, including the amygdala, really well known, but also a region that is a little bit less well known, the bed nucleus of the stria terminalis, that is really important for anxious apprehension and anxiety. And I just want to illustrate this, some of the findings here. And we did this kind of network analysis, and when we kind of show here in this force layout, we see the configuration of this network here with the salience nodes in red, the executive nodes in green. And so there's some kind of clustering here, as you can see. But the important thing here is not exactly this clustering. It's what we wanted to quantify. And this figure here just illustrates it visually. But what we wanted to quantify was the differences in network and graph theory measures, such as efficiency, and betweenness or modularity between the organization of these systems when you are in the safe condition versus when you are in the threat condition. So what we found was that in the threat condition, for instance, there was a decreased efficiency. In other words, in the threat condition, there's less clear-cut synchronization or coordination of the pairs of nodes in that network. There was decreased efficiency in the salience and in default networks. And so again, as I said, the idea then is that the sustained threat is associated with sort of like a decreased signal coordination that typically defines these networks. These networks are in fact detected because they're highly correlated. But what we find is that under the threat condition, they become less coordinated. Okay, so that was just a brief example that I wanted to show how these networks reconfigure now in the case of threat, as I had illustrated to you a second ago in the case of reward. Just the final word here about networks and labels, something that several of us have been working on for a couple of years in a OHBM sponsored task force, to try to maybe put some order into these labels and understand what we mean when we say, is what I mean by salience network and executive network and default network the same as you? But one of the take-home messages for me that I want to drive the point here is not necessarily how we should define them. It's even at a more meta level, which is, I think that we need to recognize, and it's important to recognize, that when we call a salience network, it's really involved in a lot more than salience processing itself. It's one of its functions might be related to salience, but there's a lot more going on. And likewise, when we talk about the default network, it's not just that it's involved in autobiographical memory and self-related processing. There's quite a lot going on. So I think that we have to be really careful in the way that we use these network labels, because when we use a network label that has a specific label, we might start thinking that it works for salience, but we should be thinking the same way that we should be thinking as brain regions as multifunctional, in my view. Networks are a level higher, but we also should be thinking them as quite dynamic and multifunctional. Okay, so let me just wrap up. I'm really sorry about this mouse here is just going all over the place today. My keyboard was not functioning, so it's a little non-smooth. I apologize for that. But okay, so let me just in just 2-3 minutes just wrap up here, because I'm over my time. So one way to think about emotion and cognition in the brain is to think in terms of dual processing models, in which you might have a cognitive stream and an emotional stream that interact relatively little, perhaps a little bit. There's maybe a third motivational, reward, appetitive, positive stream that is, again, separate. My suggestion, based on these studies and the frameworks that I have developed and I didn't discuss here today, is that we really shouldn't be thinking in terms of these separate systems. We really should be thinking in terms of their interactions. Complex behaviors, to understand complex behaviors, we really need to understand their interactions and not just keep them segregated. So my proposal is that one way that we can think about the brain basis of these large mental domains that sometimes we tend to separate, cognition, emotion, motivation, and so on, is really in terms of large scale and in fact dynamic cortical-subcortical networks. And the brain architecture itself, the anatomical pathways that exist, that are quite exuberant, as we discussed in the very beginning of the talk, are the ones that provide the basis for the distribution and integration of these signals that we might want to call perception, cognition, and so on. It's fine to call them that way, but there's so much distribution and integration that at some point we have to really be a little bit more careful in how we use their labels, or at least not be viewing them in a strictly segregated fashion. So the kinds of modeling, the kinds of frameworks that I would promote, that I would encourage, are ones in which cognition and emotion, motivation, action, and these other domains are much more closely intertwined. And I'm just going to wrap up here, but if you're interested in some of these ideas and more of the details and specifics, I described a few years ago a view of the emotional brain in terms of this kind of network perspective, and how this large scale network spanning the entire neural axis from cortex to the brainstem are important for us to understand them in terms of how they're functionally integrated systems that support complex functions. And as Sami mentioned, if you're interested, there's a version of this that is more broadly accessible that was published last year in this book, a relatively short book, The Entangled Brain. And with that, I have to thank all my collaborators, the funding that supported all this work from the NIH, which is in the National Institute of Mental Health, for which I'm hugely grateful. And thank you for your attention.